Good morning students, welcome to video classes. Myself Venkatra, senior faculty in botany. Today I am going to explain microsporogenesis. You already know about the structure of the anther and the structure of the microsporogenesis. Today we are discussing about the microsporogenesis. So what is meaning of the microsporogenesis? Microspore means microspores. Genesis means generation or formation. So when the anther develops, the cells of this sporogenous tissue undergo meiotic cell division to form microspore tetrad. Cells of this sporogenous tissue undergo meiotic cell division to form microspore tetrad. Each cell of the sporogenous tissue is capable giving rise to the microspore tetrad. Already you know what is a sporogenous tissue. The sporogenous tissue is a homogeneous tissue. It is centrally located in the each microsporium. The potential cells in sporogenous tissue is known as the pollen mother cell or microspore mother cell. Pollen mother cell or microspore mother cell. See here, this is one pollen mother cell, PMC, and gives rise to 4 microspores. These are the 4 microspores. The process of the formation of microspores from pollen mother cell or microspore mother cell through the meiotic cell division is known as the microsporogenesis. So that is definition of here microsporogenesis. The process of the formation of microspores from the pollen mother cell or microspore mother cell through meiotic cell division is known as the microsporogenesis. See here, the pollen mother cell or microspore mother cell is a diploid condition. The microspores is haploid. Totally here, the 4 microspores are arranged in a clusters that is known as the microspore tetrad. See completely here, the 4 microspores are arranged in a cluster forms in tetrad that is known as the microspore tetrad. These 4 microspores are commonly enclosed by the callus layer. Total these 4 four microspores are enclosed by the callus layer. At the initial stage, these four microspores are enclosed by this callus layer. And later, this callus layer is dissolved by the action of callus enzyme. This enzyme is secreted from the tapetum. You know already the tapetum is the inner wall of the anther. The inner wall of the anther. So when this callus layer is completely dissolved, these four microspores are separated. Each microspores are broken and differentiated into the pollen grains. That's why the pollen grains are developed from the pollen mother cell. This entire phenomenon is known as the microsporogenesis. See here, the total four microspores are formed from the pollen mother cell. So finally, these four microspores are functionally, each microspores go and differentiated into the pollen grains. Except some cases, you take best example, Cyperaceae family. In this family, only one functional microspores are developed from the pollen mother cell. Only one functional megaspore are developed from the pollen mother cell. So, except this family and the remaining all families, the four functional microspores are developed from the single pollen mother cell. See here, already we have discussed about this, the structure of the microspore tetrad. At the initial stage, these four microspores are enclosed by the callus layer, and later this layer is dissolved by the callus enzyme, and finally these microspores are separated from the tetrad. But in some cases, these microspores remains united. So such type of the pollen grains is known as the compound pollen grains. Compound pollen grains. In these compound pollen grains, the four microspores are remains united, so they cannot separate it from the microspore tetrad. What are the examples here? The compound pollen grains. Examples: Jatropha, Typha, Genus, and Cryptostegia. These are four examples for the compound pollen grains. Next one, in some cases, all pollen grains in each anther lobe remains united and form the sac-like structures. So such type of structures is known as the pollinium. What are the examples for the pollinium? Orchid and calotrophes. This is a milkweed. In orchid and calotrophes, in these two plants, all pollen grains in each anther lobes remains united and form a sac-like structures that is known as the pollinium. See here, the structure of the pollinium. This is one pollinium. This is another pollinium. These are a group of pollen lines. polonium. This polonium are united, a common junction is known as the translator by a star-like structure. A star-like structure is known as the caudicle. On either side of the translator, a disc-like structures are there. This entire apparatus is known as the translator apparatus. So generally this translator apparatus are present in these two plants, that is here, especially here, Heliotropics. A group of pollen grains that remains united in each anther lobe, that is known as the polonium. In some cases, see here, this one, the 4 microspores remains united in tetrad condition, that is known as the compound pollen grains. Here, all pollen grains in each anther lobes remains united. The sac-like structure is known as polonium. So these are two very important for the NEET entrance examination: polonium and compound pollen grains. What about Cyperaceae family? In Cyperaceae family, only one microspore developed from one functional microspores are developed from pollen mother cell. Next one, these microspores are arranged in tetrad is mainly 5 types are there. Let's discuss about that 5 types of the microspore tetrad. First one, tetrahedral, isobilateral, decussate, T-shaped and linear. The arrangement of microspores in a tetrad is mainly 5 types: tetrahedral, isobilateral, decussate, T-shaped and linear. See here, the tetrahedral this is a tetrahedral, isobilateral, decussate, T-shaped and linear. These are totally the 5 types the microspores are arranged in tetrad. This entire total 5 types, the most common type is the tetrahedral. It was found in dicots. Next one, isobilateral. It was found in monocots. This is the arrangement of the microspores in tetrad. The most common type of microspore tetrahedral. It is found in dicots. Isobilateral. It is found in monocots. Next one. In medical competitive entrance examination, based on this microsporogenesis, some numerical questions is asked. So that is how to calculate the number of pollen grains from anther or stamens or entire plant. See, for example, the number of pollen glands. The number of pollen grains, how to calculate the number of pollen grains from Anther. See, one simple formula is there for how to calculate the number of pollen grains from Anther. The number of microsporogenesis into the number of pollen mother cell into 4. For example, to take one flower Anther, Datura, this plant belongs to the Solanaceae family. In Datura, the total number of microsporogenesis, it is the tetrasporogenesis. Total 4 microsporogenesis are there. That is number of microsporogenesis 4 into the number of pollen mother cell. For example, each microsporogenesis contains 25 pollen mother cells. Already you know each pollen mother cell gives rise to 4 microspores. So that is the one Anther, the total number of the pollen grains. 4 means the number of microsporogenesis, tetrasporogenesis, the number of pollen mother cells in each microsporogenesis. Each pollen mother cell gives rise to 4 microspores. That is 4 into 25, 100 into 4, total 400 pollen grains. Next one, you take one more flower, hibiscus. The hibiscus flower is best example for the monothecus, only that is a bisporangiate, only 2 microsporogenesis are there. Each microsporogenesis, for example, that is contain 25 pollen mother cells into 4. Total here 200 pollen grains. In first grade, total 3 families listed: Fabaceae, Solanaceae and Liliaceae. These 3 families, that is known as the tetrasporogenesis. So regarding that 3 families only is given in further competitive entrance examination or NEET entrance examination. And next one, the number of pollen glands. The number of pollen grains, flower. The number of, how to calculate the number of pollen grains in a flower? First, the number of microsporogenesis into the number of pollen mother cells into 4 into the number of stamens. This one. Total how many stamens are present in flower? Same, you take same example, Datura. The number of microsporogenesis, how much? The number of microsporogenesis, that is total 4. In each microsporogenesis contain, you take same example, 25 pollen mother cells, it is variable. Next one, 4, each pollen mother cell gives rise to 4 microspores into. Now, the number of these stamens. Here, Datura belongs to the Solanaceae. In Solanaceae, the plants, each flower contains the total 5 stamens. That is 100, 400, that is total 2000 pollen grains. Next, the number of pollen glands. on plants. How to calculate the number of pollen grains on the plants? See here, the number of microsporangium into the number of pollen mother cell into 4 into the number of stamens into the number of flowers. The number of flowers. To take same example, Datura, the plant is contain, for example, 10 flowers. See, 4 tetrasporangiate. The number of pollen mother cell, for example, 20 pollen mother cell into 4. Each pollen mother cell gives rise to the 4 microspores. The number of stamens, 5. Each plant contains 10 flowers, 10. That is 20,000 pollen grains. So this is a very easy method how to calculate pollen grains from anther or flower or entire plants. Okay. So next one. One more important point is that the number of meiosis cell division, are equivalent to the number of pollen mother cells. The number of pollen mother cells are the number of pollen grains divided by 4. So these are two easy equivalent to the number of meiosis cell division. For example, the number of pollen mother cell 25. Automatically here the number of meiosis cell division is also 25. For example, the number of pollen grains 100 divided by 4, that is 25, the meiosis cell divisions. Okay, next one. Regarding this microsporogenesis, one more important point is that is the polysporing. So already you know, so the total 4 microspores are present in tetrad, but here the polysporing means the total more than 4 microspores are present in tetrad. That condition is known as the polysporing. The best example, Cuscutum. It is one of the parasitic plant. In this plant, so one microspore tetrad contains 11 microspores. See here, in single microspore tetrad contain 11 microspores, that is more than 4 microspores. Such type of condition is known as the polysporing. Okay, so this is the concept of the microsporogenesis. So keep watching and keep learning. Okay, bye.